Hi students, and today we are going to continue the questions from the same unit, the biomolecules, and today we are going to see the questions we can expect from the topic, the amino acids, proteins and enzymes. Okay, so see the first question. Amino acids are mostly synthesized from, okay, just compare the question with the second question. Amino acids are produced from, okay. So for your kind information, both the questions asked from a same year of 1992, I think so, NEET 1992. So you can easily distinguish these two questions. Amino acids are mostly synthesized from and amino acids are produced from. You have to differentiate these questions. I'll explain. Okay, so what are amino acids? As I told you, amino acids are the building blocks of proteins. Okay, so that means the proteins are synthesized from the particular amino acids. So proteins are made up of amino acids. Proteins are made up of amino acids. That means amino acids are building blocks of proteins. Okay, so amino acids, they are going to link with each other by means of the bond, peptide bond. Okay, and they will form the polymeric structure, that is called the polypeptide. And the arrangement of the polypeptide chain or a polypeptide chain is called a protein sequence. Okay, so the point you have to remember whenever you are thinking about amino acid, you have to remember the macromolecule protein. So proteins are made up of amino acids. Proteins are made up of the building blocks of the amino acids. So the question over here, the amino acids are synthesized from. That means I told you. So while we are studying the metabolism, I told you there are two reactions, synthesis and the digestion. Okay, so catabolism and anabolism. So over here it is talking about the synthesis. That means the synthesis of amino acids. So as I told you, for the synthesis of proteins you need amino acids. So I told you. So for the protein synthesis there are so many molecules involved. As I told you, it starts from the DNA. It carries the information for the synthesis of the DNA, and the information is being copied as RNA. There are three types of RNA, mRNA, tRNA and rRNA. So the mRNA carries the message for the synthesis of proteins. The tRNA transfers the molecules required for the protein synthesis. That molecules are amino acids. tRNA transfers the amino acids to the protein synthetic site, and rRNA will form the ribosomes. And the point you have to remember over here, from where the particular tRNA transfers the amino acid to the protein synthetic site. Okay, so imagine a cell. Okay, so the protein synthetic site, it may be the rough endoplasmic reticulum, or it may be in the cytoplasm in the free ribosomes. So the point you have to remember over here, the protein synthesis happens at the site ribosomes. Okay, so if the ribosomes present in the cytoplasm, it happens in the free ribosomes. If the ribosome present in endoplasmic reticulum, the protein synthesis happens at the particular rough endoplasmic reticulum. Okay, so the point you have to remember over here, for the synthesis of proteins, you have to transfer the amino acids from one place to another place. From which to which? Normally the amino acids are being synthesized all over the particular cytoplasm, or it is being digested from the food material ingested by the particular cell. Okay, so that kind of synthetic or synthesized amino acid is being transferred to the particular ribosomes to synthesize the particular protein molecule. So what is the question over here? It is talking about the synthesis of particular amino acid. Okay, from where it is being synthesized? What is the precursor molecule for the particular amino acid synthesis? And see the choices over here. Okay, so volatile acids, it is nowhere related to the particular amino acid synthesis. Fatty acid, as I told you, amino acid, they contain the compound called amino group, carboxylic acid group, and there are functional group along with the hydrogen atom at one side. Okay, so that structure does not fit with the particular fatty acid. Okay, so there are two other choices for you, mineral salts. Okay, there are no relationship for the salt with the amino acid. So the answer for you over here, alpha ketoglutarate acid. So I will explain something more for you for this question. So we all know, So what is alpha ketoglutarate acid, from where it is being synthesized? So in the metabolic pathway, okay, in the metabolic pathway, for example from the pyruvate, it enters into the Krebs cycle or TCA cycle. From there it will synthesize alpha ketoglutarate acid, and the particular alpha ketoglutarate acid is being act as a precursor molecule for the synthesis of amino acids. Okay, so whenever you are talking about the synthesis, you are gonna synthesize the amino acid, you have to remember the term alpha ketoglutarate acid. Okay, so come back to the second question. Amino acids are produced from. So this is somewhat confusing part of the question, the term produced. So as an animal being, we are ingesting so many different kinds of food molecules. Okay, for example, we are ingesting carbohydrates, we are consuming oils as lipids. Okay, we are consuming, of course, so many proteins. Okay, so what is the need for protein over here? From protein we are gonna get some molecules required by the particular animal cells. Okay, so what happens over here? The proteins, so whatever macromolecule ingested by us in the form of food, whatever macromolecule ingested by us in the form of food will be enters into the digestion. So what will happen over here? The digestive enzymes gonna act over here, and it will digest each and every type of the polymeric structure. If it is a carbohydrate, what will happen? Polysaccharide will be digested into the monosugars. That monosugar, for example, the glucose will enter into the glycolysis, and from the glycolysis it will synthesize or it will produce into, for example, you started off with the carbohydrate polysaccharide. Okay, it will enter into the digestion. From the digestion you will get the monomeric sugars, for example glucose, and the glucose will enter into the cycle glycolysis, and from the glycolysis we will derive the energy in the form of oxidation reduction reaction. So from the glycolysis it will produce pyruvate. From the pyruvate it will enter into the Krebs cycle, as I told you. From the Krebs cycle it will enter into the electron transport chain. From the electron transport chain we will get the energy in the form of ATP molecule. This is how our digestive system works. Okay, so we are ingesting some food molecules in the form of carbohydrates, proteins, lipids and some other macromolecules. From that we are getting the energy as a final product. Okay, so the point you have to remember over here, our question is amino acids are produced from. For example, we are ingesting a protein as a food. We are consuming egg or cereals. What will happen? That protein will be digested into smaller molecules, monomers of the particular protein. So as I told you in a previous question, the proteins are a polymeric structure of amino acids. Okay, so the proteins are made up of building blocks of amino acids. That means polymers of the amino acids will make up the structure called the protein. So if you digest the particular protein, what you will get? You will get the amino acids. That is what you have to remember. Okay, so there are two types of amino acids, or there are two classes of amino acids: essential amino acids and non-essential amino acids. So non-essential amino acids means the body itself can produce the particular amino acid. Essential amino acids means the body has to consume the particular amino acids in the form of food. Okay, so over here your question is amino acids are produced from. Okay, so you cannot produce amino acids from fatty acid, you cannot produce from alpha keto acid. Don't confuse with the term alpha keto acid. Over here the term alpha ketoglutarate acid. Okay, so alpha keto acid refers to any acid which contains the particular keto group. Okay, so this is not answer. You are not supposed to get the particular amino acid from the essential oils, because essential oils belongs to the lipids or fatty acid group. So the answer over here, you have to choose the particular proteins. Okay, so now you can differentiate these two questions. As I told you, amino acid synthesis is from alpha ketoglutarate acid. If you are gonna degrade the particular proteins, you will produce amino acids. Okay, so this is the difference you have to remember in case of the amino acid synthesis and production. Okay, so see the third question: the enzymes having slightly different molecular structure but performing identical activity. Okay, so we have seen something about the enzymes in the introduction classes. So now we will recall something about the enzymes. So there are so many different terms you have to remember about the particular enzymes. Holoenzyme, apoenzyme, coenzyme and isoenzyme. So what is holoenzyme? So as I told you in the introduction classes, enzymes are normally made up of so many subunits. Okay. Enzymes, as I told you, most of the enzymes are proteins. Just remember, not all. There are some nucleic acids, they can be act as a enzyme, but 99% of, apart from the particular ribonuclease, okay, apart from the particular, as I told you, the ribozymes. So apart from these ribozymes, most of the enzymes are made up of proteins. That means they are made up of the particular amino acids polymer, polynucleotides, polypeptides. So the point you have to remember over here, there are so many polypeptides will make up the particular functional enzyme. So each polypeptide is called as a subunit of the particular enzyme. Okay. So there are so many subunits will make up the particular enzyme structure called holoenzyme. So there are some other term for the holoenzyme is conjugate enzyme. So in other words, we can explain the term holoenzyme. For a particular enzyme, that enzyme will require some other group or some other molecule is being attached to the particular enzyme. Then only it will act as a functional enzyme. Okay, that means we can divide the particular holoenzyme into two groups or two sections. Holoenzyme, one is simple protein part. That means that is made up of only amino acids. Plus there are some other non-protein part may be present with the particular enzyme. Okay, plus some non-protein part. Okay, together we are calling this structure as holoenzyme. This simple protein part or made up of amino acid part is called as apoenzyme. Okay, so what is apoenzyme? In a holoenzyme, the protein only part is called the apoenzyme. Okay, so just remember, apoenzymes cannot act as a functional or fully functional enzyme. It needs the particular non-protein part to be attached with the particular apoenzyme, then only it can act as a particular functional enzyme. Okay, so what is holoenzyme? Holoenzyme is a multi-subunit complex. Okay, there are two regions in the particular holoenzyme, protein part and non-protein part. The non-protein part may be organic and or inorganic. Okay, so the point you have to remember over here, apoenzyme refers to the protein part of the particular enzyme. Non-protein part may be organic or inorganic. So we are calling this non-protein part as a cofactors. So already we have seen there are three classes of cofactors in our previous classes. Okay, coenzymes and metal ions and prosthetic groups. Okay, so what are the three groups? Coenzymes, prosthetic groups and mineral ions. We will call this as metal ions. So you can see it over here. Coenzymes are organic part, prosthetic group is a non-protein part, and metal ions are just elements. They are going to act as a coordination. They are going to make the coordination bonds with a particular substrate and product along with the enzyme. Okay, and these three groups are belongs to the category cofactors or the non-protein part. It is being attached to the particular apoenzyme to make the holoenzyme. Now come back to the question. You have a question, enzymes having slightly different molecular structure but performing identical activity. So what and all we have seen? Holoenzyme. There is no relationship with the explanation given in your question. Okay, we can eliminate the holoenzyme. Apoenzyme is related with the holoenzyme. Coenzyme related to the holoenzyme. So we can eliminate the three. Then the choice will be the isoenzyme. So what is your question? Enzymes having the same or enzymes having slightly different molecular structure, identical activity. For example, in a human cell, there are enzymes called lactose dehydrogenase. There are so many different variants of the particular dehydrogenases present. At least 20 to 40 different types are the variants of the particular lactose dehydrogenase is present in a particular single human cell. Okay, so what are variants? They are made up of different amino acid sequence. Different amino acid sequence. So as I told you in the introduction classes, you have to remember the protein structure is based on the amino acid sequence. Okay, so each and every type of each and individual amino acid they have in the particular polar or non-polar property, and based on that property only they will attain a position in a particular protein chain, and based on the position only the protein chain will get the particular structure, and based on the structure only they are going to act as a particular functional molecule. Okay, so over here you are having the different amino acid sequenced proteins. Okay, so that as I told you they are having slightly different molecular structure. Okay, over here what you have to remember, different amino acid sequence, different structure, but they are having identical activity. All the particular structures they are having the same function, identical. Okay, not similar, you have to remember the term identical. They are having the particular identical functions over here. Okay, and these group of enzymes are called isoenzymes. Iso means same. Okay, so isoenzymes. So what is isoenzyme? You have to remember the isoenzymes, they are the group of enzymes, they are having different amino acid sequences, different molecular structure, but they possess the particular identical functions or identical activity. So see the next question. The enzyme enterokinase helps in the conversion of something. Okay, and see the choices. So whenever you are seeing the term enzyme and a kinase, they are gonna involved in something conversion. Okay, so there are 4 choices given for you, 4 conversions. See the conversion over here, conversion of proteins into polypeptides. That means you are going to digest the protein molecules and you are going to break the particular peptides. Okay, peptide bond. So what happens over here, you are going to digest the particular peptide bond so that you will get the individual peptide units. Okay, so this is what given over here, proteins into polypeptides, digestion of protein, the large proteins into individual subunits or individual units of the polypeptides. And see the next choice, pepsinogen into pepsin, trypsinogen into trypsin, casinogen into casein. The thing you have to remember over here, from the inactive form to the active form, pepsinogen into pepsin, casinogen into casein, trypsinogen into trypsin. Okay, so all are related with one thing, one common thing, proteases. Okay, so what are proteases? The enzymes which can digest or which can act on a enzyme, and which can act on proteins. Okay, proteases can act on proteins and it will produce product from the particular protein. Okay, so casinogen is an inactive form of casein, it will be converted into casein. Pepsinogen will be converted into pepsin. But the question over here is enterokinase. Okay, so the another term for the particular enterokinase is enteropeptidase. So the enterokinase and enteropeptidase is always related with the trypsin. Okay, so they are involved in the conversion of trypsinogen into trypsin. So the choice over here, the second one, the trypsinogen into trypsin. Okay, so these are the questions you can expect from the particular amino acid segment and the enzymes. And now we are going to continue with the same segment, the questions you can expect from the same topic, the amino acids and the enzymes and the proteins. And see the next question, the role of enzyme in a reaction is, okay, there are 4 choices for you, decrease activation energy, increase activation energy, as an inorganic catalyst, none of the above. Okay, and for this question you have to remember, you have to recall the enzyme function and how the particular enzyme works. Okay, so as I told you, what is the role of enzyme? Enzyme act as a catalyst. Okay, so by this term you cannot choose this as an answer, inorganic catalyst, just because the catalyst, the word is present over here, because enzymes are organic molecules. Okay, enzymes not a inorganic molecule, enzymes are organic molecules, so you are not supposed to select the particular in C. Okay, so automatically you can eliminate the answer D. Okay, enzymes are organic molecules. They are catalyst. They can act as a catalyst, or they are catalyzing the reaction from the substrate to the product. So you are having the substrate, you are going to synthesize the product with the help of enzyme. Okay, so what happens over here, as we have seen in the last class? So from an enzyme, so towards the, so over here we are having the energy during the rate of reaction or the progress. So from the substrate, you are going to produce the product. Okay, so I told you there are two levels of energy you have to remember: stable and unstable. Stable and unstable. Just remember one thing, both substrate and products are in a stable form. Okay, both substrates and product are in a stable form. But during the transformation, during the biotransformation from the substrate to the product, it will form the unstable complex or unstable form, that is called the enzyme substrate complex or the enzyme product complex. Okay, so both the complexes are unstable. Okay, so what happens over here? First, how the process will happen, what are the steps involved over here? The enzymes go and binds with the substrate, or the substrate diffuses into the enzyme molecule. It will form a enzyme substrate complex. Okay, and the enzymes will changes the making or breaking the bonds. That is what called breaking or making of bonds. Okay, so that is what we are calling as the enzymatic action. So what enzymes are doing over here? The enzymes will change the substrate into the product. How it is changing? Initially it will form the enzyme substrate complex, then it will changes the bonding nature of the particular substrate. It will make or eliminate or break the particular bond, so that it will form a completely different structure from the substrate. That substrate, that product, that is called as a product. That different structure, formed structure is called product. Okay, so that is how the particular enzyme works. So the point you have to remember over here at the energy levels. So over here the energy is at very low. Over here the energy is at very low. So that only we are calling this as stable form. Whenever you are having whenever the particular molecular structure having a higher energy, that is unstable. That is what you have to remember. Whenever the particular molecule in its lowest energy possible state, that is called stable form. Okay, so substrate and product is in a stable form, and it has to transfer the particular unstable form. Okay, I told you unstable means it has higher energy. Okay, so this higher energy state, to attain the particular higher energy state, that particular substrate needs some energy. That energy we are calling it over here the transition energy. Okay, so we are calling it as the term transition energy. To attain a particular transition energy, it needs some activation, so that we are calling this as activation energy. Okay, so this is what you have to remember. We are talking about the activation energy. Okay, so if the substrate is in need of higher activation energy, it will take so much of time to convert it into the product. Okay, so what enzyme has to do over here? The enzyme has to reduce the amount of energy to activate the particular transition state. Okay, so what is transition? Suppose you are entering into a makeup room. Okay, suppose you are as a human being entering into a makeup room. So the makeup room is called as a transition period. So within a makeup period or makeup room, you are getting some makeup, and after the makeup completion, you are gonna come out as a better personality or better looking man. Okay, so what happens over here? The transformation happens within the particular makeup room. Okay, so while you are entering as a substrate and coming out as a makeup or the better personality product. Okay, so the point you have to remember over here, what happens during the makeup period or within a makeup room. Okay, you cannot be sit within the makeup room for some days or some hours. Okay, you have to come out of the particular room within a whatever possible time, whatever minimum period of time. Okay, so that is what happens during the enzymatic action. The enzymatic catalysis, what it tries to do, it will try to decrease the particular activation energy, so that it will reduces the time in the particular transition period, so that the product formation will be faster than the non-catalytic reaction. So come back to the choices. Decrease activation energy, increase activation energy. Okay, there are two choices for you. Okay, so the choice will be the decrease activation energy. Why? So that only it can decrease the transition period, so that only it can decrease the time in the particular transition period, particular unstable period, so that it can speed up the reaction from the substrate to the product. So the choice over here, it will decrease the activation energy. So see the next question. Some enzymes which are associated in converting fats into carbohydrates. We should have some knowledge about the particular conversion of fats into carbohydrate. So just remember one thing, from the normal pathway, in case of animal being, we are normally consuming the food in the form of carbohydrate, that is polysaccharides, for example rice. Okay, so from the polysaccharide we are going to synthesize as I told you, the monosugars, for example glucose. From glucose we are entering into the glycolysis metabolic pathway, then pyruvate, Krebs cycle, electron transport chain, then energy. Okay, so this is what the normal steps for the energy synthesis for any animal cell. Okay, from the consumed carbohydrate polysaccharide, it will synthesize the monosaccharides. From the monosaccharide glycolysis, from the glycolysis Krebs cycle or TCA, then electron transport chain, then energy synthesis. So the question over here, the synthesis of carbohydrates from fat. Okay, so when there are no enough carbohydrates available in a particular animal body, so what will happen? It will try to synthesize the particular carbohydrate from the available fat. Okay, so that is what happened over here. So the straight question over here, okay, what is the exact location of the particular conversion, fats into carbohydrate? Okay, so there are four choices over here: Golgi bodies, liposomes, microsomes, and glyoxysomes. Okay, Golgi bodies, as we all know, as we have seen in the last chapter, Golgi bodies are involved in the modification of lipids and proteins, not in a synthesis of carbohydrates. Okay, so you can eliminate that. Microsomes and liposomes are nowhere related to the synthesis of the particular carbohydrate. So the choices over here, the automatic choice over here is glyoxysomes. Glyoxysomes, as I'll tell you something about the glyoxysomes. Okay, glyoxysome is a kind of organelle. Okay, so it directly involves in the synthesis of carbohydrates from the fat. Okay, so as we all know, from the fat, okay, it will enters into the cycle called glyoxylate pathway. Okay, so just remember one thing, glyoxylate pathway is the alternative pathway for the Krebs cycle. So Krebs cycle, from the Krebs cycle normally it will enters into the electron transport chain, but over here what happens over here, from the fat with the help of glyoxylate pathway, okay, it will synthesize the carbohydrates. Okay, so what are the major needs for the particular glyoxylate pathway? There are some specific enzymes need over here. Okay, so that is what mentioned over here, some enzymes associated with the conversion of fats into carbohydrates. So what may be the role of the particular enzyme? They are going to involve in the beta oxidation. You have to remember the term beta oxidation. So what is beta oxidation? Beta oxidation is the enzymes, okay, with the help of enzymes the fats are converted into carbohydrates. So where it happens? It happens in the pathway glyoxylate pathway. What is the site of the particular glyoxylate pathway? The organelles glyoxysomes. Okay, so you have to remember the term glyoxysomes, which contains the enzymes for the glyoxylate pathway. What is the role of the glyoxylate pathway? It is going to do the beta oxidation of fatty acids or fats into the carbohydrates. Okay, and see the next question. Which one of the protein is most abundant in the animal world? Which protein is most abundant or enormous in the animal world? So there are 4 choices for you. So the easiest choice you will pick insulin and hemoglobin. We all know our body will have a circulatory system, and the circulatory system will have a RBCs, and RBCs, the matured RBCs is filled with a protein called hemoglobin. So we may think hemoglobin as a abundant protein in a human body or in any animal body, but the answer is wrong. We may think insulin as a abundant protein. That is also wrong, because insulin, as I told you, insulin involved in the metabolism of carbohydrates, especially the sugars. Okay, so if it is it should be in a particular quantity. There must be some specific quantity should be available for a particular animal being. If it is more than a required amount, that is called hyperinsulin. Okay, that will cause a disease to the particular animal being. So you cannot choose the answer insulin. Insulin cannot be abundant in a particular animal. Well, insulin required. Insulin is a must protein for animal, but it must be in a specific quantity. It must be in a minimal quantity for the particular animal being. Okay. So see the next choices: trypsin and collagen. As I told you, trypsin is a kind of the protease. It is going to involved in the digestion of proteins. Trypsin is present in our digestive system. So what is the role of trypsin? It is going to involved in the digestion of proteins. It is going to degrade the particular enzymes. Trypsin is a protein molecule or enzyme molecule. What is the action of trypsin? It is going to act on proteins, and it is going to digest the proteins into polypeptides or into some other monomers of particular polypeptides. Okay, so you may think trypsin as an answer, but that answer is also wrong, because trypsin also available in a very minute quantity in our digestive system. So the answer over here you have to choose the collagen. So the collagen is the enzyme, collagen is the protein present in a muscle cells and all the fibers. Okay, so they are the building blocks of most of the tissues. So the collagen is the most abundant protein present in a animal world. See the final question. The enzymes are not found in. Okay, see the choices: algae, fungi, cyanobacteria and viruses. And you may get confused with the terms cyanobacteria, fungi and algae. Just remember one thing: all the living cells, they possess the particular enzyme groups. Okay, all the living cells, they are having the structures called enzymes. If there is no enzyme, then we cannot call that particle or that structure as a living cell. Okay, so you can easily eliminate algae, fungi and cyanobacteria, because they all belongs to a normal living organisms. Algae, fungi, cyanobacteria, they all having the particular structures called enzymes. So the choice over here, viruses. Okay, so you have to recall the thing about the virus. So viruses are the particles. Okay, so they are normally in a inactive form, inactive particles. Okay, so when they will get activated, once they are enters into another tissue. Okay, in a living tissue only they can be act as a dividing particle. Okay, once it enters into a biological living tissue. Okay, but the particular enzyme, they may possess the genes. They may having the particular genes for the proteins or for the enzymes, but as a structure within a virus there will be no enzyme. That is what you have to remember. In a virus there are no enzymes, but they can have the particular, they may have the particular genes for the synthesis of the viral enzymes. Okay, so when the synthesis will happen, once the virus will infect the particular living cells, within a living cell, by using the living cell's own protein synthetic mechanism, okay, this virus will synthesize the particular enzyme. Okay, with the help of enzyme, it will make the particular virus particles so that it will multiply within the particular living cell. Then it will lyse the particular living cell and come out and infect, or it will enter into the other infective cycle. So the point here to remember over here from the question, enzymes are not found in as a cell algae will having the particular enzyme, fungi will having, cyanobacteria will having, but as a structure, viruses, they will not have the particular enzymes, but it will synthesize the enzymes once it enters into the living tissues. Okay, and these are the questions you can expect from the particular enzyme topic. In the next class, we are going to see something about the questions we can expect from the carbohydrates and lipids.